So Dr. Athma Pai is a geneticist and RNA biologist working at the interface of functional genomics and computational biology. Athma received her PhD in human genetics from the University of Chicago, completed a Jane Coffin Childs postdoctoral fellowship at MIT, and assumed her current position as an assistant professor in the RNA Therapeutics Institute at the UMass Chan Medical School in January of 2018. Her lab integrates both experimental and high-dimensional data science approaches to study the speed and accuracy with which our cells naturally create mRNA molecules with the spatiotemporal specificity necessary for cellular function.
Great. Well, thank you, Leah, for that introduction, and thank you also for the invitation to speak to you guys today. I'm going to do a little bit of an overview on the work that our lab does and how it connects back to precision medicine. And although I will say from the outset that we are a very basic fundamental science lab, so we really aim to use inquiry and questions about fundamental biology to understand how we might be able to gain information for therapeutic applications or even just better understand how our cells work that could come in handy in the future. So today I'm going to tell you about, as Leah alluded to, how efficiently our cells actually create mRNA molecules. And I think, thankfully for me, these days, mRNA needs less of an introduction than it used to. But I will say, and you'll see this during my talk, that I'm going to go into a little bit more depth about mRNA that's naturally produced from all of the genes in our cells. And there are various different cohorts of mRNA that are produced in different ways. So the specific topic you're talking about is really balancing.
If I can interrupt just a moment, we're getting a lot of echo and reverb on the audience side. Is it possible you have two microphones on, or can switch to a different one?
Do not, but I can see if I can switch to a different one. All right, how is that? A little bit better?
Not quite.
Okay. Let's see here. Third one I can switch to. Is that one better?
Oh, that is extremely quiet.
Okay.
And I don't think it's much better on the reverb.
Okay. Let's see, I have not had this issue before. I'm not quite sure what's going on.
Well, if those are the three possible microphones, we'll go with either of the first two that weren't very quiet and see how it goes.
The only other thing I do, if you give me a minute, I can switch to my phone.
Sure.
All right, you have to give me a second to actually dial in for this. So
yep.
Bear with me. Okay, how is that? Can you guys hear me? And does that sound better?
That is much better. Thank you.
Okay, great. Apologies, I'm not quite sure what's going on. And I have actually not given a Zoom talk in several months, which I thought was a good thing. Apparently,
that's okay.
But
technology just waits until you're in front of an audience.
Exactly. All right. Okay. Well, let me jump right back in after my apologies again for that technical difficulty. Okay. So there we go. That's great. All right. So at the core, as Leah mentioned, I was trained as a geneticist and a genome biologist, which basically means I spend a lot of time thinking about how our cells derive information from the DNA that is in all of our cells, and really is a set of instructions for creating different functions that our cells perform, allowing differentiation or developmental processes to occur, allowing cells to respond to different stimuli, or even just regenerate and grow and function in the ways that our bodies need them to continue being alive. And so at first pass, a genome looks something like this, but with 3 billion letters. And so the primary question that many xenobiologists like myself have been really interested in is, how does the cell actually find the set of instructions it needs to perform the sets of functions it does, and how does it do it in a dynamic way such that different cells, our brain cells versus our skin cells, are actually totally different entities, but still encoded by the same genome. So within our genome, there are certain features that cells intrinsically look for. The primary one, and obviously the most famous, is these entities called genes, which are the ones that actually ultimately will encode mRNA molecules and go on to create proteins or to create other regulatory molecules that are necessary for our cell. But the levels and the compositions of those genes, as I'll get into more in a minute, are really regulated by these other features called regulatory features that are not actually generated into mRNA molecules, but are still crucial for how our cells function. And while this is not quite how the proportions I have on this screen, really only 5 to 10% of our genome is comprised of genes. And instead, a lot more of it is either these regulatory sequences, of which we've probably only really begun to hit the tip of the iceberg on identifying them, but also other buffer sequences that really play a role in maintaining the efficiency of how instructions can be derived from our genome. So these sequences really encode a series of sets of information. So the genes themselves will have what I like to think of as interspersed words, and those words are what we can get sentences from, and those sentences are really the instructions that get taken to other parts of the cell to really produce proteins that drive a lot of the cellular function. And then these regulatory sequences have these motifs within them that tell the cell how much of that gene should I be creating. And there are various sets of motifs and combinations of motifs that exist usually right before the gene sequence. And these sets of motifs can be used in different combinations. You can, in particular cells, use 2 of these high motifs and create a lot of the RNA from this top gene here. Or in a different cell, you can use the 2 low motifs and create much less of it. So this is really kind of what forms the central dogma of molecular biology, where DNA, which can self-replicate, but DNA is transcribed into RNA molecules, which are then translated into protein molecules. And these proteins are really some of the key crucial actors of function in our cell. Although we're learning a lot more about how mRNA and other RNA molecules themselves can function to regulate different cellular functions as well. So this is kind of a very simplistic overview of what actually is going on. In reality, there are a whole series of mechanisms that are involved in each of these steps. And each of those mechanisms are very different from a molecular perspective and also from a spatiotemporal perspective. They happen in different parts of our cell, and they happen at different times in the life cycle of either a DNA, RNA, or protein molecule. So today, as an RNA biologist, I'm really going to focus on mechanisms involved in processing mRNA and getting it to the right place, getting it to the right levels in the right place that's necessary for cellular function. And so some of the core mechanisms that are involved in this are transcription itself, which is the process of generating RNA molecules from the set of instructions in DNA. mRNA splicing, which I'm going to get into more in a minute, and that's the process of actually reading those words from the whole sentence that's actually being transcribed from the gene. And then processes like 3' end processing, which is to say, well, when do I actually add the period at the end of that sentence? And then taking those molecules to the specific places they need to be in the cell to generate protein molecules or perform their regulatory function. So the first process I'm really going to focus the majority of my talk on is this process of mRNA splicing. And this is really the process of stitching together what we call exonic sequences and splicing out these intervening intronic sequences, which happens in this two-step molecular process. So this process of stitching together the exons creates the actual mature messenger mRNA molecule. And that mature molecule contains these exons, which actually have coding potential, which means they encode the protein by having a series of codons. But there's also other things that can be evolved in these exonic molecules. You can have these untranslated regions that are not actually translated into protein, but are crucial for regulating the levels and the compositions and the localization of those mRNA molecules. So this process is actually ubiquitous in mammalian or in human genes. Greater than 95% of genes undergo splicing out of at least one intronic sequence. And that's true for most mammalian genomes and indeed most eukaryotic genomes. And even more than that, this process can actually occur in alternative ways. So it's highly regulated. And so what do I mean by that? So this is one of those sentences, the gene sentences that I showed you before in the DNA form. And you can see that I've kind of highlighted some words here. And so the process of actually generating an mRNA molecule is reading this sentence, but that sentence can be read in many different ways. You can choose to start at different positions. You can choose to include different words or different combinations of words. You can choose to end at different positions in the sentence and actually add that period. And as you can see, depending on how you actually combine these series of words together, you can generate different meanings or different messages entirely. And if you think of now these words as these exonic sequences, this is really what our cell is doing. It's really reading the gene in different alternative ways to generate what we call alternative mRNA isoforms, or generate different messages from the exact same gene sentence. So of course, there's a different aspect to this, and that is that it's not just a question of reading it, but it's also actually entirely rewriting that sentence into a different form, right? So we are taking reading the DNA and then generating this whole nother RNA molecule from what we've read. And so because of that, the speed at which this is actually read, or how fast Kermit can actually type away on a typewriter, is regulated in and of itself. And the faster that Kermit goes, the more likely he is to actually make some mistakes in the context of reading, but also transcribing this particular instruction into a whole different set of molecules. And so this is something that happens actually fairly frequently and something that our lab studies. And that is that you can actually introduce mutations or mistakes into this mRNA molecule, which can result in nonsense that's created. And so maybe then the cell will say, okay, we can recognize that. That doesn't actually have any meaning. We're going to throw it out. Or you can generate mistakes that are still legible and still have a meaning, but change the meaning from the original intended meaning. And so this process is actually really a question of how accurately are those messages being transcribed. And so that brings us to the questions that we really ask in our lab, which is, we really want to understand the molecular basis for creating a different transcriptome or an mRNA composition. And how we do that is we develop and apply tools to study to measure the efficiency of gene regulatory mechanisms. And we study both aspects of this. We really think about how quickly is mRNA being produced. So that previous slide, how quickly is Kermit typing away on his typewriter, versus how accurately is that process being performed. And so we think of this as really a trade-off, where there's a trade-off between how fast you can do something and how accurately you can do something, even in any of our everyday lives. And so we think that's also happening inside of each of our cells at any given moment, and really understanding the speed and accuracy, how these mRNA molecules are generated, could really help us to understand a lot about the regulation of gene expression and how our cell functions. And so one aspect that we really think a lot about is maybe the speed or the timing of these events really influences how different forms of these isoforms are really generated. So I showed you before a whole different series of different sentences or different isoforms that could be generated from the same gene. that maybe the time you spend really transcribing each of those sentences might lead to different choices in which isoform or different sentence is actually expressed. So there are, this is now getting into the actual kind of taking away some of these analogies and showing you the actual process and how it's really even more complicated than what I was showing on previous slides. So this is now a gene on the bottom in the black here, and these are the start of the gene, the sites that actually regulate the splicing of that gene, and the sites that regulate the end or the position at which the end is actually put on that gene. The molecule that really does a lot of this work is what's called RNA polymerase II, that actually polymerizes the transcription of this new mRNA molecule. But there are a ton of different auxiliary factors, and one of the points of the slide is to show you how complicated this is, but also all of these arrows as to how these factors really interact with each other. We think a lot of those interactions between the individual factors that are regulating different seemingly independent steps are really what are driving some spatiotemporal relationships between different alternative decisions that are being made in the process of generating an mRNA molecule. And so what my lab really studies is saying, well, we need to understand all aspects of this to be able to understand how we can get different forms of mRNA. So we study the rates at which transcription can occur in various different forms, the rates of splicing and the rates of what's called 3' end cleavage, or the choice of the end of a gene. One of the main questions we really have is, what's the rate limiting step to produce an mRNA molecule? How, which step is really regulated from a temporal perspective? Is it the production of that molecule itself, or transcription, or is it some downstream choice that's made to express a different set of exons or different set of sites that would lead to a different alternative isoform? So one of the way in which we do this is really through an integration of various different techniques. We do what we call high throughput RNA biology, which means we really start with, well, one thing that's not on this slide is we start with a real biological question that we want to answer, and we address it through various different, both experimental and computational methods and iteration between those methods. We conduct many different types of experiments to generate large-scale data sets, really looking across the entire genome rather than focusing only on one gene at a time. We analyze these data using a lot of data science techniques in a kind of using methods from a field called bioinformatics. We then will use both simulations to simulate how we think data should look like, and these actual analyses of real data to computationally and mathematically analyze and model the probabilities of observing certain events given certain molecular phenomena. And so we use a lot of techniques from statistical modeling and machine learning in the process of doing this. And as I'm showing these arrows, we really iterate between, we kind of go back and forth between how do we better make these models and how do we better model the data, and how does that modeling and that computational work really inform the experiments that we conduct, and how is this really, how can we better design the studies that we're doing? And then at the end, we will then take a lot of the predictions that we make from our high-scale, high-dimensional analyses and really validate some of our hypotheses or really test some of the biological hypotheses that we have. Okay, so the number one technique that we use in our lab that I'm going to spend a couple minutes describing because it's crucial for all the downstream, for everything else I'm going to tell you about from a biological perspective is high-throughput RNA sequencing. And so, as you probably guessed by now, we do everything on RNA. We really try to think about, how can we sequence the entire composition of RNA to get an understanding of how much RNA there is, but also what are the different types of RNA molecules, both from the same gene and also across different genes that are really being expressed in a given cell at a given time. So if we have a pool of RNA here, this is how this technique works, is we have a pool of RNA that we can get out of a population of cells that we can culture in the lab or from samples that we get from various different sources. The first thing we really want to do is we want to enrich for mRNA, because as I mentioned, we're really interested in the creation and processing of mRNA molecules as instructions for our cell. So one of the key features of an mRNA molecule or a mature messenger mRNA molecule is that it has these marks that actually differentiate its terminal ends. So there is a cap, quite literally called a cap at the very beginning of the molecule, and what's called a poly-A tail at the end of the molecule. And what we can actually do, because it's specific to mRNA molecules, is we can use an experimental technique to specifically enrich for that poly-A tail, and therefore enrich only for those mRNA molecules that we really care about across many different mRNA molecules, because that's what's kind of common between them. And so what we do is we hybridize them to complementary string of Ts that are bound to a magnetic bead, and then use a magnet to just literally pull upon that string. And because this reaction of hybridization between these A's and T's is really strong, we're able to really selectively pull away all the RNA that has this poly-A tail and get rid of all the stuff that we don't really want to look at. So that's called poly-A selection. We get all of our mRNA. We can then fragment the mRNA into smaller pieces and make into DNA fragments, which it turns out is actually quite crucial for conducting any sort of sequencing reaction that you might hear about these days. So we can make it into DNA fragments, and on these DNA fragments, we have these little adapter sequences, and those adapter sequences allow us to go from the ends and actually hybridize them to what's called a flow cell. And so I'm not going to go into huge depth on this slide, but this is kind of the principle of high throughput sequencing. And this is specific to a technology called Illumina, but the principle is the same, where we can use those molecules or those sequences that we've added onto the ends of the DNA molecules from our RNA to really use them as anchors to start our sequencing reaction, to anchor them to a solid support, and sequentially go through, now that we've like really solidified it and immobilized it, we can go through and walk through that DNA molecule and figure out which bases are different bases and what order they occur in. That'll give us millions of short fragments. We can actually sequence, at the moment, the highest ability we have is 2.2 billion of these short fragments can be sequenced in one go, that I think takes about 24 hours to finish sequencing. And we can then take those millions of short fragments that can be anywhere between 50 to 300 sequences or base pairs long, and we can map them to the genome and figure out, okay, which genes do they come from? Which genes have more reads or fewer reads? And how do we use that information to understand the levels at which these genes are being expressed in the cell? Since we're interested in splicing, we can actually, for a specific gene, go in and look at which exons have more reads and which ones have fewer exons. And by using these reads that actually connect across multiple exons, we can say, well, what exons are connected? And that tells us information about whether or not this intron has actually been taken out, and or this entire exon, as is indicated in this top right here, has been entirely skipped or dropped out of the sequence, or dropped out of the mRNA molecules it's derived from. So this information is really useful for getting mRNA expression levels and mRNA splicing patterns, which tell us a lot about the levels and compositions of mRNAs that are expressed in any given cell type. Okay. So this technique has been around for probably about 15 years now, and it's been widely used to study the expression levels and mRNA splicing patterns in a lot of different cell types, and we've learned quite a lot from that. We've learned a lot as a field about how the composition and the levels of mRNA can change in across different tissue types, across different human individuals, across different developmental or differentiation conditions, and also, as I'm highlighting on this slide, across different types of simulations or disease conditions or perturbations to the cell, many of which naturally occur. And what's really interesting is that we know that different parts of the gene are more likely to be responsive to different stimulations. So for example, chemical stimulus really seems to affect changes in alternative usage of exons at the very beginning of the gene, while treatments like immune stimulus or heat shock or DNA damage, which can be caused by things like radiation, really seem to affect more things that are in the middle of the gene, while cancer and immune stimulation and DNA damage also seem to affect things at the very end of the gene as well, or the end of the mRNA molecules as well. And so this is really telling us that different processes have different impacts on the level to which a cell can respond to each of these different stimulations. But also, if you think back about what I said was one of our primary questions, then maybe these represent the rate-limiting steps
for how different genes are actually regulated in response to a very fast-acting stimulus, as many of these are, or even longer-term stimuli where you need to actually change the levels or compositions of mRNA in a cell to be able to respond or survive whatever that stimulation is. Okay, so as I mentioned, we're really interested in this question of rate limitation, and in order to do this, we really have to take a more temporal approach. We have to think about, as I was talking about on this previous slide, the timing at which each of these stimulations occur and the timing over which these mRNA changes could occur. And so because of that, the technique that we use is takes RNA sequencing, or that high throughput sequencing method that I showed you, a step further, because that only really sequences mature or stable mRNA. It doesn't really tell us anything about the intermediates that occur across a temporal progression towards a final state. So instead, what we want to do is capture what we call nascent RNA, which are molecules that are both before and after degradation and before full maturation. So we can kind of see each of those decisions or mechanisms that occur across an mRNA happening in real time by taking a fine-tuned time course across the timescales at which they occur. So the way in which we do that is we use a molecule called 4-thiouridine, which is an analog of a normal base pair or nucleoside that can be inserted into mRNA. And if we put that into our cell dishes for a short amount of time, 4-SU will be included instead of a normal uridine molecule, which is the analogy to thymine in RNA molecules. Putting this 4-SU in will allow us to kind of label all the RNA that's been generated within a very short time point, and we can use an enrichment technique to attach a molecule to that 4-SU molecule and actually specifically pull it down and sequence only the labeled or newly created fraction of RNA. So we do this for very short labeling periods, 5, 10, and 20 minutes, which really allows us to think about, at the scale that we think our cells are really responding to different stimuli or even turning over and changing the mRNA population that they have in them, can we really capture all these intermediates? And then finally, we do the same type of sequencing I described, this Illumina high throughput sequencing, to get millions of short reads specifically from this newly created RNA fraction. So as I mentioned, we do this across a very short time scale, 5 minutes, 10 minutes, and 20 minutes of labeling. And one thing that we expect across this time scale, and this is specific for splicing, is that as we look at increasing time, we will see more evidence of molecules actually undergoing the reaction we care about, in this case splicing, where we will see more introns being spliced out of the molecule and fewer molecules that still have introns in them. And so what we can do is we can actually say, well, let's go in and look at the rate at which that occurs. And that is how we measure the speed of any of these processes. So the data I'm going to show you very briefly today is actually showing this in fly cells, but we're now starting to do this in a lot of human cells, and this is with 5, 10, and 20 minutes of 4-SU, and that's really the time scale at which we're looking at. So this is an example of how we visualize these data, and also what this looks like for a few real genes in the fly genome. So this, let me walk you through this. In gray here are these exons that are being spliced together or stitched together, and in purple are these intronic regions that are being taken out. And so what's really nice is the darker colors are earlier time points, and the lighter colors are later time points. And in yellow, at the very top, you can see here, is what happens if you look not at a time point, but just in the full population of mRNA, most of which represents mRNA that's being generated hours ago. And so what you can see is that with increasing time, you see fewer and fewer of these intronic molecules. And what that means is that that intron is being slowly taken out or spliced out of that mRNA molecule. And you can see this for these two different introns from two different genes in the Drosophila genome. And what you can also see is the levels of exons don't really change, which is what we'd expect, because those exons are actually being retained, spliced together, and created into mature mRNA molecules. And so what I'm showing you on the right side is really just a summary of that. But now just looking at those junction reads that I told you are informative, because they can tell us about what those molecules, how those molecules can be spliced together. So in pink are those junction reads that are indicative of unspliced molecules, and in blue are the junction reads that really show us connections between these exonic fragments. And what we can see is we have a greater proportion of those spliced junction reads across time, and that's true for both of these examples. So what we can do is we can really delve into information we can learn from this. And so what I'm showing you is the distribution of the half-lives, and that's how we measure the rate, the half-lives of splicing across a distribution of intron lengths. Because one of the first thoughts we had is, to go back to this, maybe the length of these introns, so this is 63 base pairs long, and this is 686 base pairs long in the Drosophila genome. And I'll caveat one thing by saying the Drosophila genome is much, much, much shorter than the human genome. It's only about 300 million base pairs long, which means that it's orders of magnitude different in terms of the general scale of how things can work. But most of the molecular functions are still very much the same. But anyway, one of our first hypotheses was, well, maybe the length of that intron impacts how fast that intron can be taken out. And so what we looked at was, well, is there any correlation between the half-life or analogy to the rate of splicing and the length of that intron? So I'm showing you here, this is the distribution. Each point is one intron here. And in blue is a running median across this to show you just generally, what does this look like across it? And you can see there's actually really not very much correlation. So our hypothesis was entirely wrong. There is no overall correlation between the length of an intron and the rate at which it's taken out. But what's really interesting is you can look right here, there's a very predominant dip in the half-lives right around 60 nucleotides. And what's really interesting is if you look at the mode of the distribution or the median, analogy to the median of how the distribution of intron lengths, greater than 30% of Drosophila introns or fly introns fall between 60 to 70 nucleotides. And we also know that there's evolutionary pressures to really favor shorter introns. So this is a distribution that's very different than the human genome, where the median length of human introns is about 1,000 base pairs, not 60 base pairs. But what's really interesting is that we see this dip in the rate of splicing right around, even if we zoom in, right at the 60 nucleotide range, where even 10 base pairs greater has a slightly longer half-life or slower rate of splicing. And when you get shorter, you also have a slightly slower rate of splicing. So what this tells us is there might be some sort of selective pressures, evolutionary pressures, to really push intron sizes into what we think is the most efficient size for taking out an intron, and that there are some constraints, either biochemical or otherwise, that really mean there is an advantage for introns to be about 60 to 70 nucleotides because they can be taken out and spliced quicker and more efficiently. So I'm not actually going to go through this, but I'm going to skip to this and say that we learned a lot of other things. This is actually a published paper on splicing. We learned a lot of other things about how the architecture of a genome may actually be determined by how efficiently the rates of each of these processes is actually occurring. So we extended this method. On the left, I'm showing you the distribution of splicing. On the right, now I'm showing you the distribution of the rates or half-lives for 3' end cleavage or polyylation. And that's this decision to put the sentence on the end of the molecule or choose the very end of the molecule and say, okay, I'm done. I have my full mRNA now. And so the choice of these positions also happens at some rate. And what we can see is that this rate is a little bit faster than the rate of splicing, where the median rate of splicing is about 1.92 minutes, while the median rate of cleavage is about 1.25 minutes. So they're not too far off, but they're happening very, very quickly as these molecules are being transcribed. And so one of the things we're really interested in now that we're starting to get to and really working on in collaboration with a few other people at UMass is to really think about, well, how can we think about the gene as a whole rather than just individual introns or individual terminal ends, as I've been showing you so far, but instead, how can we really understand the rate at which these processes are occurring across the entire gene? And to get to therapeutic applications or to get to really an understanding of how our cells see this, we have to think about the gene as a whole, or the entire molecule, entire mRNA molecule that's being created, because at the end of the day, it's not an individual exon that matters, it's how those exons perform in the context of the other exons that they're connected to. And so we can actually go through and measure the splicing rate or the cleavage rate for different sites, then we can think about if you have what happens in this gene, two different possibilities, two different isoforms that can be read from the same gene. And each of these intron or 3' end decisions has a particular rate. How do we figure out what the rate limiting step is for generating each of these? And is there actually competition? Can you say, well, this happens faster, so therefore you're more likely to get this site because it happens faster, or maybe depending on how long it takes to get to the site, this site, even though the actual cleavage event happens slower, it's been around for longer. And so therefore it has just as much of a possibility of being cleaved at this bottom site as this top site. So anyway, this is stuff that's very much in progress, really understanding the dynamics of how this works. Okay, so that was about one half of what my lab works on, which is really thinking about the speed of mRNA production. But as I told you at the beginning, we're really also interested in the accuracy, or what are the mistakes our cells make. And I think this is where we can really start maybe seeing some of the more immediate connections with precision medicine and how this might have therapeutic applications, because the decisions as to, when are the mRNA molecules that we're actually generating the proper mRNA molecules are really important. Because if we have a mutation in one of those mRNA molecules, there can be drastic consequences for the cell. So the way we start to think about this is actually because I told you that we do a lot of nascent or newly created sequencing of RNA rather than this mature RNA sequencing. And we realized that because of the fact that we're sequencing intermediates in the process of generating RNA molecules, we're also actually sequencing a lot of mistakes that might otherwise get removed in the process, in the context of the cellular function, right? So as I mentioned before, there might be an mRNA sentence that's created that's completely nonsensical. And the cell says, well, I know that's nonsensical, I'm going to get rid of it, right? That sentence is definitely something that I can copy edit out. And so therefore, we don't really see it in the final population of RNA. So that scenario described of copy editing, the cell performing copy editing, is not that much of a jump. There are a lot of what's called proofreading mechanisms that the cell actually undergoes to do just that, to remove mistakes in either the DNA replication, the mRNA transcription, or the protein translation process. But what we wanted to do is we wanted to say, well, let's actually understand some of those mistakes or some of those intermediates that can tell us more about the mechanisms and tell us more about perhaps some of the sites that might be mistakes that can either tell us what's going wrong in the disease or allow us to use some other information to try to reset a disease on the right path. So we use our nascent RNA sequencing to really sample these intermediates, and we can sample a number of different types of intermediates. And today I'm going to tell you about two of them. The first is recursive splicing, which is actually not a mistake, but a way in which a cell can really efficiently splice out, we think, really, really long mRNA molecules. And the second are these cryptic or low fidelity splice sites, which are really more mistakes. They're things that are not really meant to be used, but represent sequences that are similar to what can be used and therefore are used at a certain rate and can be cleared out of the cell, but can also be harnessed. Okay, so first I just want to show to you that we realize that we can actually identify these because a lot of people usually look at what's called only annotated or canonical splicing. They don't really think about, well, what are all these things that are mistakes, that are splicing events that are happening to sites that don't usually get used. And that's what I'm kind of showing here in red. And so this is from a paper I was involved in a number of years ago that we realized that the longer the intron, the more of these unannotated sites you could actually pick up if you went and looked for them. We were not able to find very many of them because, as I mentioned, they're really hard to find with traditional RNA-seq data sets. So we realized that using our really time-resolved RNA-seq data sets would present an advantage and could really build upon the work that we had done 10, 12 years ago now. So the first is this idea of recursive splicing. So recursive splicing is really allowing the cell to break up a really, really long intron and say, well, I don't think I can take that entire chunk of the genome out in one piece. And these are often 100,000 base pairs that it's trying to remove from mRNA. Instead, it's going to use an intervening sequence to chop up that intron into small pieces and take it out in segments rather than as one whole. So we developed a computational method to really identify places in which this was occurring, and we identified that longer introns tend to have more recursive sites, and there are some interesting patterns as to how they're positioned in the genome that perhaps indicate that they're regulated and they're on purpose rather than totally randomly existing. But one thing that we noticed, which is really interesting, is that this process of actually recursively splicing out an intron creates this this kind of sequence beacon in the middle. That is what the cell wants the machinery to use. And because of that, it is perhaps less likely to use random, similar, but not quite right nucleotides in the middle. And so what we realized is that if we can actually quantify the proportion of inaccurate or kind of noisy cryptic site usage in these introns that show recursive splicing versus introns that don't show recursive splicing, we can understand whether or not this beacon that's in the middle of the site is actually performing any function in allowing for more accurate splicing. And that's exactly what we see here on this plot, where I'm showing that introns that are just as long as introns that have recursive introns or recursive sites, but do not have recursive splicing, actually tend to have a lot more inaccurate splicing than introns that are recursive spliced. So we think that this is a mechanism of non-canonical splicing that actually allows for or promotes accuracy of splicing with the entire molecule. So the second thing, the very final thing I want to tell you about is the idea of how do we actually go look for these inaccurate sites that are cryptic sites used potentially by mistake by the cell. So this is actually an example of a gene. This is called SLC6A2. It's a neurotransporter gene that it turns out this and other family members do have many mutations that can lead to developmental delays and seizure disorders in young children. And so when we look at this gene, we're kind of curious, well, you know, how accurately is the splicing of this gene? Because that might have impacts on how much this gene can be expressed. So we can do some computational predictions, and that's what this track is here, to say, well, where are all the potential sequences that are close but not quite to the sequences that are actually used, or to the ones denoted in the boxes. And what you can see when you do computational predictions is you can see hundreds of these sites sprinkled throughout the gene. Now the question is, well, if you want to go in and actually target some of these sites to try to block mistakes from happening, I don't know how to prioritize any of these 100 sites. But when we add our actual data on top of it, and that's what I'm showing in the curves here, in black are the ones that we expect to happen, and those are good. Those are the ones where the cell is regulating the usage of different alternative mRNA synthesis as it should. And then we look for all these other mistakes, which are the ones in red. We can now start to see that they line up. If you look very closely, they line up with the computational predictions, but they allow us to really subsample these computational predictions into ones that are actually being used. So this data can really tell us a lot about, well, what are the places where mistakes are more likely to happen? How can we use that information to understand how mistakes happen? And then also, how can we actually block these mistakes from happening? And so this is experiments that are in progress. We can actually use these oligonucleotides that are called antisense oligonucleotides, small stretches of RNA molecules that can bind to the RNA and block the site from being used. And our hope is that can actually promote an increase in the levels of the productive or good forms of this mRNA from being produced, especially in children that have mutations that might decrease the levels of this or any other gene. So just in the interest of time, I'm going to skip ahead to show you, this is just what I mentioned, okay. So this is another gene that is much more complicated in its structure, right? So now it's not just a question of pulling out these 10 sites that we can maybe target and we can really just grab a handle on, but instead on the top in red here are the regions that have more cryptic sites than the ones in black here. And so now what we're in the process of doing is we're really developing more fine-tuned algorithms and analysis techniques to really allow us to say, well, how do we actually even further subsample such that we can more intelligently figure out where can we direct our therapeutic investigations to among all of these splicing mistakes that I'm showing on the top here, so that we don't end up just brute force targeting hundreds of sites, which would not would kind of defeat the purpose of doing these experiments in the first place. And so this is a gene called NF1, which is really important in Friedreich's ataxia and different diseases like that. And so this is also a gene that is very commonly mutated and is something that is of a great therapeutic interest to figure out, well, how can we kind of leverage this information about mistakes in splicing to try to correct or increase the levels of genes. And so this is just a diagram that shows again that what we hope to do is really force the cell to express the corrected forms of the mRNA or the proper forms and really redirect the products of what we think is wasted transcription from these cryptic mistakes towards productive transcriptional outputs or mRNA molecules. So with that, I'd like to leave some time for questions and thank everybody in my lab. And this is the actual the building on the UMass Medical campus that we work in, and this is it nicely lit up at night, kind of right at the heart of the campus if you ever drive by our campus in Worcester. And so with that, I'm happy to take any questions. Hello.
Okay, so just to start off with one question, you mentioned at the very beginning that your process involves a lot of sort of big data analysis. And I was wondering is there any culture of data sharing or data reuse, or does every lab sort of have to produce its own individual data, or is that something in the future that could sort of help increase efficiency if there was data sharing?
Yeah, that's a great question. So I think we're very lucky that the genomics community from the very beginning has actually adopted a very much more open access to data kind of mentality, perhaps in other communities. But so all data that we publish, high throughput data or even smaller data, and including any scripts or kind of computational software that we develop, has to actually become publicly available at the time of publication. And so the reason I pulled up this slide again is that this figure was actually generated from data we did not generate. So this is actually an experiment that was conducted in this paper on the bottom here, Journal of Molecular Biology in 2015 in a cell type that honestly my lab doesn't even have. So we couldn't be able to conduct this experiment, but we were able to download the data and actually analyze the things that we were interested in. They looked at totally different things in these same data. And so yeah, I think this is definitely something that a lot of us promote and kind of have taken advantage of in the past and continue to do so.
That's great. So we have an audience question. This was an interesting mix of biological and computational techniques. How do you train your students and trainees in both of these very complex areas?
Yeah, that's a great question. So my lab is, I'd say now about 50% experimental and 50% computational, and we really have a distribution of people who fall along that spectrum. So some people are 20% experimental and 80% computational and vice versa, and some people really fall right in the middle at 50-50. And I think that there's two aspects to answer your question more directly. Is that one, I think that people who join my lab tend to be more interested in actually integrating these two aspects. So oftentimes they will come more experienced in the experiments and have a great interest in learning in the computational stuff, or they come more experienced with a background in computational biology and have an interest in learning the experiments. And one of the things that I really heavily emphasize is that modern biology is really going in a very quantitative direction. And because of that, I think integrating those techniques in the same lab is becoming increasingly necessary. In many, I didn't have time to really go into the details of these, but in many of these experiments, if we didn't know how we were going to analyze the data, we probably would have designed the experiment incorrectly. Or if we didn't know how we generated the data, we probably would have analyzed the data incorrectly because of, as you mentioned, a lot of these complex kind of decisions that are made while both doing the experiment and doing the analysis. And so, so I think that, you know, there are many resources on the UMass campus in terms of classes and different labs that have taken this approach and therefore have really emphasized this integrative learning to answer a biological question rather than just gain a set of skills.
Thank you. I have another follow-up question. So as the theme of this conference was personalized and precision medicine, how do you see the concepts of precision medicine sort of developing within your own work, either in current directions or in future hoped projects?
Yeah, so I think, as I mentioned at the beginning, a lot of what we do, and as you saw, was really fundamental questions of, you know, how does biology work? But I think that, first of all, you know, before you get to precision medicine, you have to kind of know what is standard versus what should be more specific to individuals. And so I think one of the things that really high throughput data can tell you, both across, you know, different cells and different individuals, of which we do quite a bit of kind of sequencing samples from different individuals to learn the degree of natural variation across individuals. I think one of the things you set the baseline for, well, what is abnormal versus not, right? And so in a medical context, I think that's really important to understand. The second thing is what I'm highlighting on this slide, and that is that a lot of these kind of harnessing the mistakes in splicing for therapeutics perspectives, we're actually collaborating with Jonathan Watts in his lab on campus that work on oligonucleotide RNA therapeutics. And a lot of these techniques, our ultimate goal would be to really use for individuals that have rare mutations that downregulate or decrease the levels of a particular gene. And because it's a rare mutation, it's something that can't really be treated by some existing common drug therapy, because it's very much more specific to that individual. And so, so one of the things that we really see as a precision aspect is that these oligonucleotide sequences, these very short sequences that we use to block the usage of any of these mistakes or block these mistakes from happening, can be designed quickly in a more precise fashion to particular individuals. Now it becomes very, very precision medicine where you're saying, well, this individual has this mutation. I'm going to design an oligonucleotide drug that's very specific to that individual and then use it to regulate the levels of mRNA that's being produced. And this is not something that's just a pipe dream. This is actually something that has been done a few times now, both by my colleague John Watts and others, both in the Boston area and otherwise. And one of the most famous examples is a drug called Milasin that was designed for a young child named Mila. It's a story that I encourage you to go look up, where they actually designed an ASO, this antisense oligonucleotide, to redirect a splicing pattern, very similar to what I'm talking about, towards increasing the levels of expression of a gene that was causing the disorder that she had. So one of the goals that we have is to say, well, you know, we want to contribute to this. However, one of the real bottlenecks is that when people go to identify the sites that they can actually target to kind of correct these mistakes, the mutation that causes the disease is often not causing a splicing difference, but you have a lot of mistakes that are happening naturally in the cell, such as what I'm showing you here. And so what we hope to contribute is to say, well, maybe what we can do is we can target a different site that's causing this mistake in splicing to occur and not necessarily need to really know anything about how that particular mutation works, because that can often be a whole set of years of trying to figure out how a mutation functions. And so because of that, we're trying to really attack this again from a fundamental perspective of saying, well, how do we really understand which of these sites is most likely going to give us the most promise for increasing the levels of a gene? And how can we use that information across a set of patients or an individual patient to even more quickly design a precision drug that would allow you know, the redirection of the expression levels or mRNA levels in that individual.
Thank you. If there are any other questions, please feel free to put them in the Q&A.
I have a couple in the chat from
Okay.
panelists. So, so back on the sites of inaccurate splicing slide, what suggestions do you have for finding the data that you used for such a thing, finding data like that to reuse for your own work?
Yeah, yes. I think you're referring to these data that we use from a published paper. So there's a few different ways that we do it. The first is that we will be able to search things like Google Scholar or PubMed for papers that have worked on cell types that we're interested in. This is a lot of these processes occur in a very cell type specific manner, and so we need this is actually a neuroblastoma cell line. So in this case, we really need neuroblastoma cells to understand expression of this gene because it's only expressed in neuronal cells. So we can actually look for papers that have worked on neuroblastoma or other neuronal cell types, and then find that, and then they will always link to their published data because, like I said, it is required when you publish to have the data published. The second way is that they're actually repositories that are the repositories where people deposit data because they need to make the data available when published. And so these are actually repositories run by the NIH usually. So there's a few famous ones. One's called Short Read Archive, one's called Gene Expression Omnibus, and there are a few other ones that are European archives as well. And so we will often go to those, and they have really nice filters that allow us to say, well, can we filter by different cell types and different data types, and be able to download the data directly from that. And so I see the second question here. I think the second question is, can the splicing of RNA be used to change the phenotype or 3D structure to limit or enhance degradation or proteins, possibly to help with therapeutics for diseases like Alzheimer's or ALS? Yes, it certainly can. I think we're a long way off to understand exactly how the splicing of RNA, especially for more common diseases, can be used to change the phenotype. We know that splicing is very dynamic and changes in different cellular conditions, different cell types, and certainly in different diseases. And we know that those regions that are not translated can actually help to determine the translation rates or stability of RNA or the stability of proteins. So there's a lot of work actually going on to understand splicing in neurodegenerative diseases like Alzheimer's or ALS. And one of the reasons is because splicing is very, very diverse in neuronal cell types. So neuronal cell types exhibit the most alternative splicing. So they will have the most diversity of different mRNA sentences that can be read from different genes. And so we're not quite sure why that is, although maybe that is because the neurons are very, very complex. But I think that leads to a lot of areas to research, but also unfortunately leads to a lot more complications for getting a direct answer on how to target different aspects. And I see another question in the Q&A.
Yeah, we had a request for if you could just repeat the name of the drug that was created for the specific rare mutation.
Yeah, so I'll type it in here. It's called Melicin, and this is a story that was actually, I want to say, 4 years ago now, maybe 3-4 years ago now. Actually 3 years ago, I think it was in 2019. Yeah, there it is. Yep, so Millis, Miracle, and Melicin. And so you'll get probably the whole story and links to the paper and articles. And unfortunately, Mila herself has actually passed away now, but it is still a very interesting example of the potential promise of some of these therapeutic avenues, especially as we start to learn more about how these therapies can work and again, where to target these particular therapies. Of course, you know, this is not, I think, precision medicine, as most of you probably know, is still still very much in development. And so I think this is a good example of a case where it certainly showed promise, but not really understanding other complexities of the disease and the progression of the disease kind of hinder the use of one drug or one set of drugs to really completely ameliorate a particular condition. But it's, from a potential promise of some of these oligonucleotide therapeutics avenue, it's a very interesting example that certainly showed promise.
Okay, thank you so much.
Great. Cool. Sorry. Stop my sharing. Great. Well, thank you so much for having me.
Yep. Thank you very much, Dr. Pai. I see lots of hand claps coming up, so thank you for your talk.
Thank you.
Have a great afternoon.